Next, I'd like to introduce Paula Vij from the University of California, Davis, who's going to talk about designing a CRISPRa therapeutic for STXBP1.
Good afternoon, everyone. First of all, thanks for the foundation for their support and for allowing me to have a talk today about our work in developing CRISPRa as a potential therapeutic strategy for STXBP1 disorders in patient-derived human neurons. And our main goal is to determine whether CRISPRa and restoring the healthy allele expression levels can serve as a platform that is independent, very independent. And we've heard a lot about this disorder. We are all familiar with it, so I'll just briefly talk about this introduction part. STXBP1 are neurodevelopmental disorders that which are caused primarily by the loss of the functional of one copy of the gene, which leads to decreased protein expression levels. Individuals with this disorder, they have cognitive impairment, epilepsy, and neurodevelopmental disabilities. What is interesting is that even though there's different mutations reported, most of them share this common molecular mechanism known as haploinsufficiency, where 50% roughly is being expressed only, and thus leading to these phenotypes that we see in these individuals. STXBP1 encodes for this important protein found in the presynaptic synapse, where it helps with the fusion of the vesicles and neurotransmitter release. So having the reduction of the protein, it impairs the fusion of the vesicles in the presynaptic membrane, thus leading to the impairment of neurotransmitter release and downstream signaling pathways such as communication in the neurons. Now, knowing that this disorder is fundamentally affected, or the problem is dosage, we ask the question, how can we increase the expression of the gene? And there's multiple ways of doing it. We just heard a very nice talk about the different strategies, gene replacement, ASOs. But CRISPRa is the one I'm going to focus on. It uses three main components: the catalytic dead Cas9 protein that is going to be tethered with different activators, and we target these two transcriptional regions of the genes, promoters or enhancers, with guide RNAs. Now, our hypothesis is that increasing the endogenous STXBP1 using CRISPRa can restore physiological protein levels and enable phenotypic rescue. So, just briefly, a summary of what I've just said. Haploinsufficiency is the molecular mechanism of this disorder. The goal is to increase the gene expression. Our strategy is using CRISPRa. And when we think about developing therapies, we just don't think about the effect of this, but we also want to think about the translational translational purposes. And it's known that the gold standard for gene delivery is AAV. However, one of the limiting factors is the capacity of this AAV. So in the lab, we are exploring two different systems. The first one is a dual AAV Cas9 system, where it uses the canonical and well-studied Cas9 protein. And then when we tether with the effector and the domains, it overpasses the packaging capacity of the AAV, so we split this into two AAVs. This system is very powerful. It's very modulable. However, the limitation is that the cells need a co-transduction of both vectors, which makes it difficult. We're also exploring another Cas9 system known as the mini Cas9. It uses another ortholog of the Cas9 protein. It's less studied, but it's more compacted. And so when we tether with the effectors and the guides, it fits into one AAV, which for translational purposes, it's more ideal. The downsides of this is that it's less powerful, and the PAM sites for the Cas9 ortholog is more restricted, and so it's harder to work with. Our experimental workflow, we get cells from patients with different mutations. We do a baseline characterization that then will inform us about phenotypic rescue. We screen these guides in neuroprogenitor cells, and then, like I said, we do other rescue experiments. I just realized this is the first set of slides that I sent, the first version, but that's okay. And so, so before we do any characterization or any neuronal phenotypes, we first want to make sure that our starting material is okay. That way later the biology that we see is not due to mechanical defects or any experimental problems. So we get cell lines from patients. We have 4 different cell lines that we're currently working with, all harboring different mutations that we obtain from the Simons Foundation. We confirm pluripotency across all 4 different cell lines as well as the controls. And then we take these cell lines, we differentiate them into neuroprogenitor cells. This is where we do the big, large guide screening because the neuroprogenitor cells are easier to grow, they're faster to do screenings. We also confirm their cell identity using canonical markers such as Nextin, SOX2, and PAX6. And across all the different controls and the mutant cell lines, we see comparable cell identity and morphology. When we dive into the CRISPR-A, guide design is one of the most important things for CRISPR-A success. The reason why is because CRISPR-A depends a lot on promoter structure and the chromatin availability. And so here to your right, there is like the 2 different kind of schematics of the 2 different systems that we're using where we're doing different guide screenings. And then, to your left is just a representative of the NSCs or the NPCs that we grow before we do the CRISPR-Cas screening. What we've seen so far, we tested some guides, 10 guides in the split Cas9 system, and we see that across 4 different variants, some of the guides are successful at upregulating the gene. Interestingly, lead guide 2 creates a very robust effect in 4 different cell lines. Contrary, using the mini Cas9, we see that it's not as robust. We don't see significant upregulation, and only 1 cell line has significant upregulation. And it is something that we will kind of expect, knowing the biology of the mini Cas system that is less powerful. And if we go back to this image, to the bottom is the position of the guides in the mini Cas system, and due to the restricted PAM sites of this Cas ortholog, those guides are quite a little bit far from the transcriptional star site. So we will think that that's what this minor effect. So we're currently optimizing this system, trying to do some multiplexing of guides and trying to screen different effector domains as well. Now, once we get some lead guides, we want to screen this in the disease-relevant cell type, which is neurons. And we've heard a lot of NGN to neurons. We know that it's a direct neuronal conversion from iPSCs to NSCs. The advantages is it's a very fast and reliable protocol, gives us high yields of neurons, excitatory neurons, which is a cell type that is primarily affected in this disorder, and has low variability and across different variations and inductions. These are the neurons that we have generated in the lab with 4 different mutants of the patients, the different patients. We again confirm cell identity using neuronal markers, NUA and MAP2, and then we do this for further analysis. Okay, I had a slide before this, but once we have neurons, we also do some multi-level characterization. We do morphological analysis to see how the neurons grow, the complexity of them. We also do some synapse development assays through confocal microscopy, since synapse is one of the main phenotypes that we want to see in this disorder. And we also do functional assays because we just don't want to increase the gene, but we also want to see what's the effect at the functional level. So we do calcium imaging in the lab, as well as recently acquired a collaborator using MIA recordings. I started working in this project in February. We took some time. It was some issue getting the cells in our lab. So this is just data from February, but I haven't got to the point of testing this in the neurons, but I just want to show this proof of principle data. I've worked before with another disorder, STXBP1 related intellectual disability, and we saw that doing this characterization at the morphological level, different variants have different phenotypes, which is very interesting because not just STXBP, but other disorders such as STXBP1, it's still very unknown how the variant affects the phenotype and the biology of the neuron. So seeing this in STXBP1 was quite interesting. And most recently, we also collaborated with Dr. Nadav at UCSF, and where we screened their CRISPR-Cas system in our NGN2 Pet Hopkins syndrome model. And we saw that this was successful at creating some rescue at the morphological level, at the morphology of the neurons. So this kind of gives us and paves the way for us to continue this work into the STXBP1 cells that we are currently have growing in the lab. And so in conclusion, we know and we believe that STXBP1 is an ideal candidate for CRISPR-A therapeutic strategies. We have enabled this human patient-derived platform for characterization of different variants and CRISPR-A testing and development. We think that one of our guides can have the potential to regulate this, not just at the mRNA level, but we're currently also looking at the protein level. And further optimization of the mini-Cas9 system is being done, as well as further characterization of the neurons. Overall, we think that by restoring endogenous STXBP1 using the CRISPR-A system can be a potential mutant-independent platform for as a therapeutic strategy. And with that, I just want to thank the lab, our collaborators, and as well as the foundation, and I'll take any questions.
Very nice talk.
Hello.
Thanks. More of a comment, I suppose, but I just wanted to sort of share the commiserate, perhaps. We've also tried a number of the more compact, slimmed down CRISPR activation tools, and I have to say, on a number of different target genes. And we have just never seen anything even close to as potent as the original sort of dCas9 VPR system. We've tried to miniaturize the Cas protein, miniaturize the activator, and even though they look good in a number of different publications, we've really never seen anything nearly as robust as with the sort of gold standard original, I suppose. And so I just wanted to add that comment because and it's great, it would be amazing if you guys do see sort of a breakthrough there, but we've wasted a lot of failed hours trying to use some of these miniaturized constructs. So just a perspective.
Yeah, no, that's great. We also have done CRISPR mini screening in Pet Hopkins syndrome, and we do see activation significantly, but it's not as robust.
Yeah, so I worry about this for STXBP1 though as well, right? Where we know we need pretty significant mRNA upregulation of a highly expressed gene. It seems like a long shot, one worth testing, but it would be ambitious, I think, to expect really robust, sufficiently robust activation with a weaker activator. Right,
yeah. Like I said, that's something we're optimizing, doing the multiplexing of guides. I don't know if you have tried with like the multiplexing both the syntaxin as well as the STXBP1, but that's something
we have, and we do see synergism, so that's worth exploring.
Yeah, okay, thank you.